Hello, everyone. Depending where you are, I have to greet you with good morning or good evening or good afternoon. My name is David Lee from MedChem Express, and it is my pleasure to introduce the first kickoff webinar presentation for the MedChem Express webinar series. And the webinar series with the goal of broadening scientific research and accelerating the research progress. So today, it is my pleasure to introduce our first speaker, Dr. Bart van Hazebroek from University College London Cancer Institute. So his talk today will focus on his 30 years exciting career and exploration of PI3 kinase research. And he will share the lessons learned and looking to the future. So before the talk, I'd like to make a brief introduction of Dr. van Hazebroek. Dr. van Hazebroek is a professor of cell signaling at University College London, and he received his PhD from the Laboratory of Molecular Biology at Ghent University, Belgium. After his PhD training, he carried out postdoc studies at the Ludwig Institute for Cancer Research, supported by a prestigious long-term career development award from the NFWO. After his postdoc training, Dr. van Hazebroek became a faculty member at University College London and moved to Barts Cancer Institute, Queen Mary University of London. He was then elected as a fellow of the Academy of Medical Sciences and became professor of cell signaling at UCL Cancer Institute. Dr. van Hazebroek has made key discoveries and published more than 200 articles about the functions of the PI3 kinase in cancer, inflammation, and immunity. And his research combines fundamental studies of the biochemistry and cell biology of the PI3 kinase pathways with efforts to translate these findings into diagnostic and therapeutic applications. His seminal work includes the isolation of the genes of several isoforms of PI3 kinase and the delineation of their biological roles in normal physiology and disease. So again, it is my pleasure to bring Dr. van Hazebroek to you, and let's welcome Dr. van Hazebroek.
Okay, welcome to all online. I'm just gonna try to share the screen now. It's the first time we organize this, so I need to have a quick look, share screen. Here we go. And if I now go into presentation mode, can you just confirm that you, no, I just need to swap this display, swap the presentations, or maybe not. I think that's not the right thing to do. Oh yes, I think it is. So let me just check. Do you see my pointer? Do you see the?
Yes.
Yeah. Do you hear me okay?
Yes.
And you just see slides and no, command buttons at the bottom, slide one of whatever. You don't see that. Okay, really good. Okay, we'll go. So I'm going to put my timer on. Okay, welcome to all of you online. And, thank you very much to MedChem Express for the opportunity to talk to you about my favorite signaling pathway. As is alluded in the title, I joined the field more than 30 years ago, and I will take you to this journey of PI3 kinase discovery and translation. And the field had its ups and downs, like many fields. Very early drug development to target PI3 kinase was done before all the science was known. Now there is a second wave. We understand better how the PI3 kinase pathway works, and we think we will be able to make better drugs for therapeutic application. So in this talk, it will be largely a focus on our own work and somewhat from a personal perspective. So this is a very broad field, and I apologize in advance to the many people who I will not be able to mention today in person. So let's have a look. Yes, if I go to the next slide. So here's an overview of the talk. I will give a general introduction of PI3 kinase. I do expect that not everybody is an expert in the field. Then I will talk about inhibitors of these enzymes, one isoform, one family member in solid tumors, and another one, PI3 kinase delta, in immune cells, which is really a story with surprises, as I will tell you. And then, after inhibiting PI3 kinase for more than 20 years, we have actually moved on to make small molecule activators of this family, and I will elaborate on this towards the end of my talk. So I just need to do some disclosures. I will talk about PI3 kinase delta inhibitors, and I might mention some of these inhibitors here, but I am an advisor SAB member of these companies. So how does PI3 kinase work? So here we are looking at the plasma membrane of a cell, or a membrane also in the cell. You have a receptor, an activator that can be a receptor, a small GTPase, activates PI3 kinase. And what this PI3 kinase will do, it phosphorylates a lipid in the membrane called a PI lipid. And what it does, it puts a phosphate on it. And then that lipid with the phosphate on it is recognized by different proteins in the cell, X, Y, Z. And these proteins do many different things, and they control cell signaling, vesicular traffic, and so on. So just to wrap up, PI3 kinase makes a lipid in the membrane. You have a lipid binding protein, and you get a biological output. So just go back a little bit. In the late 80s, PI3 kinase was known as a biochemical activity. This was discovered by Lou Cantley et al., who did some biochemical experiments. And then basically what's happening, this is more detail on this lipid here. This is basically a ring with phosphate, where you can put on phosphates in many different combinations. So PI3 kinase phosphorylates this inositol ring and makes it a phosphorylated derivative. Many other people were also involved in this very early stages of the work. So in 1991, 1992, the first cDNAs were cloned of PI3 kinase. And these are two seminal papers here from the Mike Waterfield lab. So this is Mike Waterfield. I joined his lab 30 years ago in October 1993, who worked closely with Peter Parker on this. So they characterized PI3 kinase, which actually exists at least for some of the isoforms as a kinase, P110, in complex with an adapter that can dock onto a receptor. So they cloned the 85 regulatory subunit and the P110 catalytic subunit. And other groups were also involved in this. So I joined Mike's lab when they had done this seminal work. And as was mentioned by David, what we did with other members of the lab, we basically cloned a lot of PI3 kinases. It turned out that they are a family of enzymes, the class 1, 2, and 3. Here are the P110s again, alpha, beta, delta, gamma. These are the ones that I mentioned. They act on the plasma membrane. They make these lipids, and they couple to cell surface receptors. They regulate a lot of proteins, and they control signal transduction in the cell. The class 2, 3 enzymes, they make lipids, but more in also in other compartments inside the cell, both the plasma membrane and the intracellular membranes, and they control intracellular vesicular traffic. They can control signaling, signal transduction, but they do it in an indirect way. So for the purpose of today, we will focus on the class 1 enzymes. So what do they do? Tyrosine kinases, G protein coupled receptors, small GTPases, they can all become activated. They recruit or activate the 85-110 subunit. This molecule is converted to a lipid. That lipid interacts with proteins. And these proteins, to give a bit more detail, some of you might know them, AKT, PKB, mTOR, adapter proteins, GTPases, and like AKT has itself maybe 100 substrates. So you can imagine that this lipid, by activating a kinase like this, controls a massive amount of biological activities. And it's not surprising in a way that if you have an enzyme that can do all these good things for a cell, that cancer cells, amongst other, hijack this, that things can go wrong in immunity, too much PI3 kinase, metabolic disease, aging, neurobiology, cardiovascular, you name it, PI3 kinase tends to be involved in one way or another. And one enzyme that I should actually also mention is PTEN. PTEN is actually carrying out the opposite reaction to PI3 kinase. So PI3 kinase puts on a phosphate, goes in this direction. PTEN takes the phosphate off. And I will come back to PTEN in a minute. So why are they drug targets? Just to recap, in cancer, they are frequently activated in cancer, especially this P110 subunit, which comes with its gene name PIK3CA. That's an oncogenic activation, or PTEN. So basically PTEN is inactivated, and so the PI3 kinase pathway is always on, not switched off. So P110 alpha and PTEN will come back in my talk. They are not only mutated and activated, also when you treat cancer cells with chemotherapy, radiotherapy, hormone therapy, targeted agents, they often become resistant, and often this resistance is mediated by PI3 kinase. So inhibiting PI3 kinase in cancer seemed like a good idea, and it is. In immunity, in immune inflammation, the P110s, two of them, delta and gamma, are highly enriched in leukocytes. Leukocytes, the white blood cells, so they are drug targets in immune inflammation and leukemia. Leukemia, the tumors of the white blood cells. So this really led to very extensive PI3 kinase inhibitor development efforts in biotech and pharma. And really this field honestly exploded. Just before we go there and talk to you a bit more about the drugs, I would like to talk to you about class 1 PI3 kinase in rare diseases. Each of these enzymes, or at least alpha, delta, and PTEN, have been implicated in inborn defects. The first one is PTEN hamartoma tumor syndrome. Basically, these are people who are born with a germline inactivation of PTEN. That predisposes them to specific cancer. The brain also overgrows, and there is a tendency for autism spectrum predisposition. PIK3CA-driven overgrowth, that's called PROS. This is due to somatic mosaic mutation early in development. And this is Mandy Sellers, the first person identified with this mutation early in development. Mandy is very happy to show this picture. She is quite an advocate for patients, and she's happy for us to show this picture. It's very clear that the lower part of her body has massive overgrowth. And then PI3 kinase delta, that's a germline activating mutation in PI3 kinase delta, leads to immune dysregulation and predisposition for leukemia. You know, also these rare diseases are obviously of interest for PI3 kinase inhibitors, because this is all activation, aberrant activation of PI3 kinase in all tissues, in some tissues, and in the white blood cells. So let's go back now. To recap, we have 4 class 1 isoforms. We call them alpha, beta, delta. So basically PI3 kinase alpha, beta, gamma, delta. These are the gene names. And in cancer, as I mentioned to you, it's mainly this PI3 kinase alpha that is oncogenically mutated. So from a cancer perspective, that was the first one to go for. You find alpha and beta in all tissues. And as I mentioned, gamma and delta are mainly in the white blood cells. So which drugs have been approved? Mainly delta inhibitors, like this one here, which inhibits delta. This one inhibits delta and gamma. This one controls delta and other PI3 kinase isoforms. This has been approved for some B-cell malignancies and also of this rare immune disorder, inborn immune disorder. PI3 kinase alpha, the most mutated, has been approved for PROS, the overgrowth syndrome, and also in breast cancer. I think what you will spot here is that, you know, maybe 100 companies worked on this PI3 kinase inhibitor development. We have 1, 2, 3, 4, 5, 6 drugs. approved. So, you know, this is really a very poor outcome. And I will guide you a little bit to what was happening here, why that is. So I'll focus, the rest of the talk will really focus on PI3 kinase alpha and on PI3 kinase delta. So if they are approved, how do they work? And so let's talk about PI3 kinase alpha first. I will come back to delta in a minute. One thing that confused the field really is, you know, obviously you have the PI3 kinase in the cancer cell and you want to block it with the PI3 kinase inhibitors. So companies jumped on this and they made inhibitors that indeed inhibit PI3 kinase, but they inhibit many different other things, many signaling pathways beyond PI3 kinase. And they also used, at least in vitro, in test tube, very high doses. So, you know, everybody thought that PI3 kinase inhibition could kill every cell on Earth, cancer cell. But, you know, we were not really looking at PI3 kinase only. We were looking at other things too. You know, the oncology field, the drugs do not always have to be so specific. A bit on off target, a bit of what they call dirty drugs can be tolerated. But I think that has, you know, at least in the PI3 kinase field, you know, made things a bit unclear. Then when companies started to make very clean inhibitors that were on target for PI3 kinase, it turns out that they don't kill cancer cells. So, you know, if you want to cure your cancer, you have to kill the cancer cell. Basically, they are quite good at anti-proliferation, so basically stop the cell from proliferating. And I think what was also, to me personally, at least surprising, that people or companies use viability readouts, cell viability readouts. But what does that mean? A cell viability readout does not measure cell death. And I'll give you one example. So this is like a lab example here that I will show you. Basically, you take 5,000 cells in a well on day 0, you see them, and you let them grow over 3 days. If you treat them with the vehicle, the control, you get 50,000 cells on day 3, okay? With the PI3 kinase inhibitor, the cell stays there, and at day 3, you still have your 5,000 cells. You haven't killed a single one. When you then, so basically you can actually achieve 90% of inhibition of proliferation, but without any cell death. And so people used MTT, MTS, cell titer glow, which is marketed as a viability assay. These are readouts for metabolic activity, not for cell death. And so basically this is growth inhibition, which is a good thing in cancer, but not killing the cancer cell. And if you use really proper cell death assays, the PI3 kinase inhibition only shows, at best, a few % of cell death. And I think for people who are interested in this, you know, I put the paper up here from the Peter Sorger group that explains this very well. So if a PI3 kinase inhibitor doesn't kill cancer cells, how can it then be a good thing to be approved in cancer treatment? Well, they're not cytotoxic, but you need to combine them with something. And so this growth inhibition, so that's the bottom line here, the growth inhibition contributes to PI3 kinase action in cancer, but requires combination to induce cell death. So let me give you an example. Solid tumors, breast cancer, the inhibitor that has been approved, it has been approved for endocrine therapy resistant mutants, PI3 cancers of the breast, which are mutant, which are hormone responsive, and HER2 negative. And this is in combination with the estrogen receptor degrader. So what is this? So you have PI3 kinase, you have the estrogen receptor. Estrogen controls breast cancer proliferation. With the PI3 kinase inhibitor, you take the PI3 kinase out, you do have the anti-proliferation that I mentioned. The cells really don't like that. And what the cell does, it starts to make more of this estrogen receptor. And it actually becomes more dependent on this estrogen. So basically what's happening is that by giving a PI3 kinase inhibitor, you stress the breast cancer cell. And what you do, you make a therapy that worked in the past, but you make it work again. So hormone therapy works more effective or becomes effective again. So, and not everybody in the field will agree with this, but in my view, PI3 kinase inhibition is a good biological modifier, but not a cancer cell killer. So I hope that is clear. if you find the right combination, you can achieve big things with a PI3 kinase inhibitor in cancer. Well, there are still outstanding issues, of course.
Me, Dr. Van Hazerbrook.
Yeah.
Excuse me, sorry to interrupt. Could you increase the volume a little bit?
Okay, I need to speak louder, I think. I need to speak louder. Is this better? Is this better?
It's a little better, yes, thank you.
You know, I am in a shared office, so, and I mentioned to my colleagues that I will be talking, but I need to speak up. So people, if you're listening outside in the office here, sorry. So, okay, are there other combinations that you could use beyond hormone therapy? You know, I showed you that PI3 kinase does a lot of things in normal physiology. Drug tolerability will remain an issue. PI3 kinase does many things. How do you target the cancer cells and not the normal cells? People try to give intermittent dosing, like with AKT inhibitors or delta inhibitors. A very exciting development is that companies are now managing to make inhibitors that are selective for this PI3 kinase mutant. So they don't hit the normal PI3 kinase anymore. They can only go for the PI3 kinase that is active in the cancer cells. And that would take away a lot of the side effects that are currently seen with this PI3 kinase inhibitors. People are also making covalent inhibitors of PI3 kinase. So basically, people are trying to avoid hitting the normal PI3 kinase and going for the mutated one. Some scientific problems, really. You know, we really don't know what this oncogenic PI3 kinase does. Is it important in the initiation of cancer? Is it important for the cancer cells to stay there? Is there really oncogene addiction? Probably not. And then there are many PI3 kinase mutations. Some may be important, some may not. And so this is what they call variants of unknown significance. You know, there are hundreds of mutations. So if a person is mutant for PI3 kinase, it may be a mutation that is not actually very important. So when it comes to sort of having diagnostics to select a person for treatment with a PI3 kinase alpha inhibitor, the person may have a mutation, but may not be an important mutation. And then cancer cell drug resistance is going to be a formidable obstacle. And that's true for all targeted therapies. You know, if you target a certain node in a cancer cell, they find ways around it. Okay, so let's move to PI3 kinase delta, my favorite, the gene I cloned a long time ago. So just a recap, alpha is ubiquitously expressed, delta is highly enriched in all white blood cells. So we cloned this gene in 1997, and we used mouse gene targeting using to find the role. So how do you go about to find out the role of a PI3 kinase? So if you look back at this picture here, if you knock out the PI3 kinase, alpha, for example, well, the others might just go to the receptor here and compensate. So, you know, we thought that's not a good idea. So people thought, I'm going to go back. Maybe take away this adapter subunit. I think that's in principle a good idea, but something odd happened. So basically take out the 85 and then that P110 kinase can no longer go there. But what happened? And again, this is a data figure from a paper shown here. We look at this lipid again, and you look in time how this lipid is produced, how much, upon stimulation with insulin, which is a classical PI3 kinase activator. This is what you see in the non-targeted cells and the wild type. Look at that. In the knockout cells, you have more PI3 kinase. You know, that's clearly not what the PI3 kinase inhibitor are going to do. So this sort of confused the field somewhat. And I can tell you many stories about this, but I think, you know, we went for another approach. So if you think about it, we wanted to model an inhibitor. We wanted to test the role of the kinase activity. So what does an inhibitor do? It hits the kinase, it blocks the kinase, but it doesn't take it away. It doesn't do it like a knockout. So if you think about an inhibitor, this is the ATP binding pocket. ATP is necessary to carry out the phosphorylation. You see this ATP nicely fits in here. This is the gamma phosphate. An inhibitor takes up that space. So how are you going to go about if you want to use genetics to model this inhibitor? Well, there was a simple trick that we applied. There is a conserved amino acid here. It's a DFG motif. You find that in all kinases. It is important to coordinate this gamma phosphate here. And so basically what we did, rather than knocking out the gene, we simply made a mutation. We converted this D to an A. And what's happening? You make this thing kinase dead genetically. So just to recap, this is not a transgene. You do gene targeting. You look at the endogenous PI3 kinase. It's not transgenic by homologous recombination. And basically the mouse is completely the same as the wild type, apart from this point mutation here. And so PI3 kinase is kinase dead. And you know, this has been a real gold mine for us. We published the first mice in 2002. In fact, this was the first kinase dead mouse ever made using this approach. So what did we see? We found that the PI3 kinase isoform that we blocked is expressed at wild type levels. We did not see deregulation of expression of the other isoforms, so things did not go up and down, as in many of the knockouts published. It models what the on-target toxicities is. So, you know, if you see a side effect, that is really due to this a function of this kinase. We found that the PI3 kinase isoforms, although they are very similar, they have non-redundant functions, so they do separate things. We have clear phenotypes in the heterozygous mice. This is actually quite important. If you think about a drug, a small molecule inhibitor, the drug will not hit the kinase all the time. When you take a pill, the dose goes up and goes down over time. So hitting the kinase 100% all the time in all the parts of the body is not a good model for drug testing. So our heterozygous mice were really much better in this regard. And so when pharmacology then turned up that people made inhibitors, we found a really good correlation between the genetics and pharmacology. And you know, I'm pleased to say that the findings we had with the kinase dead mice has really translated very well to the human context. And so one of the tricks we did, if you take a cell where the kinase is inactivated genetically 100%, and then you throw a PI3 kinase inhibitor on it, for example, you have a delta kinase dead cell, you put in a delta inhibitor. The delta inhibitor, if it's very clean, shouldn't do anything because you cannot inactivate the kinase twice. And so that's how we found out that a lot of these early inhibitors, they really changed, you know, thousands of genes in a cell that had no P110 delta. So that was quite, and I'll come back to that. So just we made, out of the 8 PI3 kinase isoforms, we made 6 kinase dead mouse lines. That's where the literature is good for. You can have a look. So let's now have a look on what do we do with PI3 kinase delta. So when we cloned this, made these mice, we got in the press, people got quite excited, thinking about immune inflammation, allergy, wonder pill, you know, that's what I say, the heights of the overpromise at some point. But there was one thing that was actually a bit strange in these mice. They developed colitis. And, you know, to cut the long story short, if you take clean PI3 kinase delta inhibitors in humans, they also get colitis. But no attention was given to that. I will come back to that. So let's have a look at B and T cells. So B cells and T cells are lymphocytes. They are your immune cells. Delta mainly controls this. Gamma controls the innate immune system. So really a lot of efforts happened. A lot of Delta programs were initiated. We collaborated with companies labeled in red here. And so the idea was, okay, the Delta inhibitor will block immune inflammation and will kill leukemic cells. So let me go back. And none of these turned out to be true. So let's have a look at B cells. We found that PI3 kinase delta is a very important player in normal B cells. And what it does, downstream of the antigen receptor, B cells recognize foreign antigens. The B cell antigen receptor signals, and it signals really through PI3 kinase, but also other kinases, BTK and SYK. Other stimuli of this kinase, of this delta, are adhesion molecules, co-stimulatory molecules, and chemokines. And what's happening? in leukemia, the gene is not mutated, and only in some B-cell malignancies like CLL or follicular lymphoma, this signaling pathway remains active. They remain dependent on that, and that really creates a specific vulnerability. So only the cells where this signal is maintained have been found to be, up to now, to be the targets of a PI3 kinase delta. Although PI3 kinase delta is expressed in every leukemic cell, apart from maybe multiple myeloma, it does not is not an anti-cancer agent in those for now. Basically, in 2014, we were very happy. A delta inhibitor was approved, the first PI3 kinase inhibitor, but really serious side effects. And it turns out, as I kept on mentioning, most inhibitors are not very delta selective. Many of them, or some of them, have really metabolized, so the drug gets metabolized by the liver, and then you can get toxicity in the liver, which has nothing to do with delta, but with the side, let's say, degradation product of the drug. And I think what was also crazy in a way, that drug developers were really focusing on the leukemia side of things and didn't pay enough attention that this delta also controls the immune system, so that you can actually not only affect the cancer cells, the leukemic cells, but also the immune system. So it was obviously a big problem. So the field really went through a bit of a dip, and to some extent for some of the inhibitors still is. So, but how does it work then? The drug was approved. If I tell you all this sort of nasty things about this drug, how does it work? It doesn't kill the cancer cells. How does it work then? Here is, one explanation for this. Look at this lady. She has CLL, which is a B-cell malignancy. She has a lot of B cells in the lymph nodes. With the PI3 kinase delta inhibitor, these cells disappear. And so what's happening, this is a waterfall plot. This is the size of a tumor, 0 or 100. Each bar is a patient. You can clearly see that in most of these patients, the tumor really shrinks to basically -100. So this is quite something in a way. So what's happening here? And I told you that PI3 kinase inhibitors are not cytostatic as a monotherapy. You need to combine them. So the delta inhibitors are approved with anti-CD20 or chemotherapy. So how does it work? And it's quite exciting actually. But this is not something you can sort of predict from some test tube experiments. So here we go again. We look at a leukemic cell that is in the bone marrow or in the lymph node. It gets all this stimuli that I showed you, especially the B-cell antigen receptor. It grows. CLL has a peculiar lifestyle. It actually goes into the circulation and then goes back. So this is a bit of a vicious circle here. And so what the delta inhibitor do, it basically, in the leukemic cells, it blocks the metabolism and proliferation. It makes the cells more sensitive to other therapies. It sort of stops all these signals from happening. So what's happening is that, as I showed you by these patients, all these cells disappear from the lymph nodes, but they actually go into circulation in the blood. This is massive. And what's happening, these cells cannot respond anymore to the chemokines that attract them to the bone marrow or the lymph nodes. They don't respond to adhesion signals. The signals are blocked, so they don't proliferate anymore. And so basically, this is a slow exhaustion. So in circulation, they are away from their protective niche and really very vulnerable. They are not getting the supportive signals anymore. And then, you combine them with something else, anti-CD20 or chemotherapy, and you can kill them off. So again, PI3 kinase delta inhibitor is an example of a biological modifier. It modifies the biology of certain tumors and it makes them vulnerable to certain specific drugs. So I think if we could find more of these, I think there is a good space for PI3 kinase inhibitors. The biggest surprise came when we did this experiment. We thought that the PI3 kinase delta dead mice, when we injected them with tumors, we were expecting them, because the immune system is maybe not working as well as we thought, that the tumors were going to grow bigger. So this is a tumor cell line that is labeled. You can measure that by imaging. In the wild type cells, you get more of the tumor. Look at the kinase dead. The tumor initially grows and then disappears. So we were very surprised about that. So there's clearly not an immune suppressed environment. How does that work? And, to cut a long story short, this is an inhibitor. It does block the lymphocytes, but there are different types of lymphocytes. Here are the lymphocytes that kill the cancer cells. Here are the lymphocytes that help the immune system to happen. And these are the bad guys. They oppose, they are the suppressive guys, they're regulatory T cells. So what's happening is that the cells that kill the cancer cell, they're a little inhibited. The helper cells a little bit, but the suppressive cells are inhibited more. So basically the immune suppression falls away, which means that after a couple of weeks, the system rebalances. So basically, this is immune activation by immune suppression. Differential impact on differential immune cells. And so we published that now 10 years ago. And this really opens the door for delta inhibitors in cancer immunotherapy, because if you can switch on the immune system, then you're in business, because that's a way cancer immunotherapy to attack cancer. The question is this obviously happening also in humans? And it is. So people went back to the immune, to the hematological malignancies. They did an analysis later. You find that the regulatory T cells, suppressor cells are blocked, and the activated T cells get up. We run a clinical trial with the Amgen delta inhibitor in cancer research, with Cancer Research UK, in a window trial in head and neck cancer. And so we basically also saw the same, that you get anti-tumor immunity, the Treg number goes down, and the CD8 killer cell go up. But there are immune side effects, which, we sort of know from the mice. So I think now that we know all these things, we can actually start to work with these drugs, cleaner drugs, better. So we know what to expect. So what's happening? One company, Iontura, has a clean delta inhibitor. They can do continuous dosing. They get the immune activation, not the tox. They are doing trials in solid tumors. They have recently been granted FDA orphan drug designation in January 2023, and they're also trialing them in leukemias. And other delta inhibitors, if you give them sustained, that can be a problem. People use intermittent dosing. Okay, the last bit. In the last 8 minutes or so, I will talk about activators. So PI3 kinase pathway has been targeted by loads of inhibitors. But, activating kinases could also be of benefit. Using genetic tricks or inhibitors, people have found that PI3 kinases can be involved in tissue repair, wound healing, neuroprotection, regeneration, ischemia reperfusion. I will come back to that. If you activate delta, you could maybe activate the immune system. You can stimulate autophagy, and so on. So basically, the PI3 kinase can be genetically activated. We asked the question, can you also make small molecules? So we focused on the PI3 kinase alpha. So we published this earlier this year. We've made the first alpha selective activator. And I'll tell you a little bit about this. This was a collaboration with AstraZeneca. We did some medchem on it, and I'll talk about a molecule called 1938. It is like drug-like molecule. It has an IC50 in the micromolar range. What is surprising to us, that it does not hit beta or delta. Look at this. Only activates alpha. It's very selective. It doesn't hit any of the other isoforms we looked at. It activates also the oncogenic PI3 kinase. And now that we have the crystals, it's very clear why this compound cannot activate beta or delta. It simply does not fit in the pocket. The pocket is obscured. I think, this is, okay, let me go back here. So how does it work? It's an allosteric activator. and we did crystallography and so on. And so the PI3 kinase has multiple domains. And it really sort of nicely snuggles in between all these domains, and it actually does a lot of things to this. It binds outside of the ATP binding site, and it blocks this inhibitory interaction that P85 has on P110. It makes changes in the active site. It repositions the kinase activation loop. It brings the ATP loop and the N loop closer together. So really, this is like a very small thing, and I compare it with the splinter in your toe. It's very tiny, but it does a lot of things. You walk differently. So, for the people who are interested, if you have a look at the Nature paper, this is a fantastic. There are videos in there how this works. It's quite peculiar. So have a look in cell lines. It induces the lipids that I mentioned to you. Here is a cell line where you add the 1938. Each line is an individual cell. You get quickly this lipid produced. If you give a PI3 kinase inhibitor, you quickly reduce it. And you don't see anything in the knockout cell line when the alpha is not there. It's actually more potent in cells than outside of the cells. When you look at knockout cells, these are mouse embryonic fibroblasts, which have wild type or no P110 alpha. In the wild type cells, you can clearly see activation of the PI3 kinase pathway in a dose-dependent manner. You don't see that in the knockout. When we look at an unbiased way, this is using phosphoproteomics, wild type cells, clearly nothing to be seen in the knockout cells. So we're quite happy with what we see here. Have we made something that will induce cancer? We think it's not the case. 1938, the signaling is very transient. It's a bit like growth factors, as you see here. We look at activation over time. This is insulin in blue and activated. So this activation is still subject to the normal downregulation phenomena, and we think it's different to oncogenic PI3 kinase. What I will not show you, it does what you expect of PI3 kinase. It activates the metabolism, it pushes cells into cell cycle, and it induces cell proliferation. Let's have a look what we can do with this. Cardioprotection and regeneration of peripheral nerve injury is what we looked at, because we were looking initially for application based on very local application and very short-term treatment with 1938. So let's start with cardioprotection. So why would PI3 kinase be involved in this? Well, let me tell you first a little bit about ischemia reperfusion injury. When you have a heart attack, the coronary arteries are blocked, and basically it is ischemia, lack of oxygen, and that induces damage. Surprisingly, when you reperfuse with oxygen, oxygen is a dangerous friend. It induces a reactive oxygen species. Basically, by the stent that is being put in under the perfusion, you get more damage. And so this is a way where you could possibly interfere. So ischemia reperfusion damage. Our colleagues at UCL had actually been working for a long time at what they call a reperfusion injury salvage kinase pathway, the risk pathway. What's happening if the heart senses stress? It can activate a group of survival kinases, MAP kinase pathway, PI3 kinase pathway. And you can actually induce that by insulin and other factors. So there is a way of protecting the heart when you put a stent in by giving insulin, for example. We actually also did studies with them that if you look at this protection and you add a PI3 kinase alpha inhibitor here, you get actually more damage. So the question really is, can we get that with an activator? And the answer is yes. So basically you take, this is a study in explanted heart, you give a global ischemia, then you reperfuse, and you give the drug 1938 alpha activator for the first 15 minutes of the reperfusion. And then you look at infarct size. This is a heart here sliced into all different slices from top to bottom. The red is live tissue and the white is dead tissue, as you can see here. It's clear that with the activator, you have more red tissues. So the infarct size has been reduced, correlates with phospho-AKT. And this is only by 15 minutes treatment. And we also have protection in live animals, which I will not show you today. So what about peripheral nerves? Just to be clear, peripheral nervous system, for those who are not familiar with this, is everything that is shown in yellow here. So we're not talking about the brain or the spinal cord. So if you have your spine broken, we're not talking about this disease setting. Peripheral nerves, they do regenerate, but they are slow and it's incomplete. So here is a healthy nerve that innervates a muscle. A muscle is damaged. A surgeon can repair it, so they put it together. And then the axons will slowly grow back to that muscle and innervate it. The problem is it's too slow. And by the time these axons have found the muscle again, it's too slow. The muscle has irreversibly degenerated and cannot be rescued. So what we think, with the PI3 kinase activated, can we speed this process up? And can we do this, get actually a faster regeneration? And so the idea behind this is actually very simple. I'm sure most of you know that there's growth factors, many people have tried that. They stimulate signaling, but if you look what's underneath here, it's actually a very simple idea. Rather than giving one growth factor or 3, you just give a PI3 kinase activator. You just enhance the music across all these growth factors. So what did we see? Well, basically, there's actually no medicines at the moment to stimulate nerve injury, and it's actually very common. We first did some in vitro studies on dorsal root ganglion neurons. You get an increase in neurite outgrowth, which you can partially block with the PI3 kinase alpha inhibitor. So this stimulated us to go to an in vivo model. This is a sciatic nerve crush model. This is a rat, spinal cord, the nerve going to the muscle. You can actually experimentally make a cut or a crush here, as you can see here with the forceps. There's a damage here. And then you can actually be injected to the activator. We put a pump in, and then you can actually look at this nerve by electrophysiology. You can actually do histology here. You can actually look how this nerve connects back to the muscle and do histology and so on. And to cut a long story short, when we look 3 weeks after injury, everything is improved. And I won't go into any detail. All the details are here if you want to look later in line. But at the histological level, at the electrophysiological level, and the functional level, these nerves regenerate much better, and the muscle as a consequence. And so the mechanism is really very simple. Everything is accelerated, nothing different. Rather than having more nerves or neurons and things growing apparently, everything looks perfectly normal, it's just quicker. And that's actually what we want, because what neurosurgeons say, time is muscle. The more quicker you generate, the quicker you have your muscle function back. So time to summarize. I hope I have convinced you that the landscape of targeting PI3 kinase is changing. With regards to the inhibitors, they are approved, so the people who have these rare diseases might benefit from that, and some really like an APDS, they clearly are. The anti-cancer mechanism, if you want to target PI3 kinase in the cancer cells, it can be used to potentiate other cancer therapies. If you want to affect things outside of the cancer cells, stimulating the immune system, as shown here, this is the cancer, this is the immune system, you can activate that. When it comes to immune inflammation, I don't think that the delta inhibitor will be good for your arthritis, because long term it will activate the immune system, and you don't want to activate the immune system in arthritis. So I think that is going to be a tricky one, unless you sort of give the delta inhibitor for a short time. There's also new developments. I did not mention this. You can use the drugs in cancer prevention or early intervention, the mutant selective inhibitors, intermittent dosing, and so on. Activators, it's a new tool to explore PI3 kinase signaling function, possible utility in tissue protection and regeneration. And, we can inhibit many of the PI3 kinase isoforms we found. So that will keep us busy for the next 20 years. I just want to sort of end a bit on a joke here. We wrote a review in Tips we got the cover. This gentleman said, stop PI3 kinase. That was 10 years ago. I think what we now need to do is to activate PI3 kinase. And maybe just a very general summary, really. Like with a lot of things, new technologies, people are really, something happens and people have this expectations and there's often inflated. Then you go to a trough of disillusionment. I think you can then go back to a slope of enlightenment and then become very productive. I think with the PI3 kinases, we are somewhere here. And if I then wrote this review that the PI3 kinase inhibitors are finally coming of age, I really believe that. So thanks to all the people in the lab. I didn't name people current and present. A lot of people were involved. We have loads of collaborators, and a big thank to our sponsors, and thank you for your attention. I think that's it. Over to you, David.
Okay. Thank you very much, Dr. Van Hazebroek. That's a very interesting and exciting talk. And so for the audience, please add your questions in the Q&A, and we can choose some of the questions and then would give that questions to Dr. Van Hazebroek. So, okay, so first question here. Has anyone tried to use two separate PI3 kinase isoform inhibitors, provided the person has overexpression of those two isoforms?
Yeah, it's a good question. I think what people try to do in the beginning, say, look, we can actually make inhibitors that hit all the PI3 kinases in one go. So we'll do that way. Developing two drugs is actually quite complicated. And that was, in fact, the sort of ambition. Overexpression, mutated, leukemia, let's just go in. And I think we're now at the stage of isoform selective inhibitors, alpha or delta. I think the stage to sort of combine them, they're all each in development separately still. I think maybe at the later stage you could sort of start to combine them, but I think it's too early days. I think they are really trying to find out how to deal with an alpha inhibitor or a delta inhibitor. I think once we know that, I think maybe you can start to do that. I think what is a good idea, and in fact, what you could argue, if you have a breast cancer that is not hormone responsive anymore, you make it more responsive with an alpha inhibitor, you could actually, after you have done that, still give cancer immunotherapy with a delta inhibitor, so sequentially. I think that's not impossible, but probably a little bit too early for now.
Okay, so a question following on that is, with the different inhibitors for different isoforms, has the outcome on the cell proliferation, inhibition, and immune response activation, are those response change depending on what isoforms you are targeting?
Yeah. I think anti-proliferation is probably quite common. So I think it's across the cell types, whether it's an alpha inhibitor or a delta inhibitor or a pan-PI3 kinase inhibitor. I didn't get the second part of the question. I think the immunomodulation you will only have with the delta or gamma inhibitor, not with the alpha, really.
Okay. Yeah. So there are a lot of questions. We may not be able to answer all of them. So, so there's another question. Does inhibition of other cancer targets like phosphatases or demethylases act as biological modifiers like PI3 kinase inhibition? I think it's a little bit out of scope of this talk.
No, but I think it's a good point, because I think when one thinks about cancer therapy, it's often too focused in my mind to only, we need something that kills the cancer cell. And I think you can change the environment, you can change how the cancer cell reacts and so on. So I think modifying how it responds to another drug, for example, an existing cancer drug, if you find, people try that for years to sensitize to chemotherapy. If you find sensitizers to therapy XYZ. You know, someone from a big pharma at the meeting we organized a month ago, he said, you know, and they worked on PI3 kinase, said, look, I think PI3 kinase is a fantastic tool to combine with anti-cancer agents as a modifier. So I think the question is well taken.
Okay. So so I think there are time for us to maybe 2 or 3 more questions. There is a question from the audience. Oh, there's okay. So one question from attendee says, do you have safety concerns with the PI3 kinase activator? And do you feel that the 1938 activator has application systematically, or would it need to be more localized administration for safety concerns?
Yeah, it's a very good question. It's a question that keeps everybody busy. Our early data suggest that systemic exposure is not a problem. Okay, so but I can't go in too much detail. Less than a problem than I had thought, you know, to be honest. We would always envisage, you know, we have several other applications in mind, and for now we would always base them on a, let's say, a 24, 48-hour exposure, not long term. That's one thing. The second thing is, you know, if you go in diseases like oncology or others, you know, this is a tough indication. You could sort of say, is this going to be cancer promoting, yes or no? Well, if a person already has lots of cancer, that is maybe a less of a concern, you know. And then I think the last but not least, the way we these inhibitors, activators turned out, you know, they don't stay very long in the serum. The systemic exposure is very low. So yeah, I think for now, because of the activation also being so transient, we don't have real concern. But for now, I think we would be looking to be careful. I think I would not give it chronically for a year, for example. You know, this is not an anti-inflammatory.
Okay. Yeah, so there are 2 questions related on patients with APDS. So one question was about the use of PI3 kinase delta inhibitors, the possibility in the patients with APDS. Would that lead to the immune activation in the long term?
Yeah, it's a good question as well. It had not been seen. So basically, these people have an overactive PI3 kinase delta, yes. And what they seem to be able to do with this inhibitor is just bring it back to, let's say, baseline level. The way the drug is dosed, you know, it doesn't go like does not inhibit the delta further. I think there's an element of normalization, not inhibition. So the way the drug is dosed is just to bring the overactive delta back to the normal level, not to a level to inhibit it.
I see.
So, you know, that's how the drug is used.
Okay. And along that same line, so another audience mentioned that in the same patients with APDS, so patients are losing the CD4 positive T cells and therefore the T regulatory cells. And do you think there's some peculiar T regulatory biology going on? And then whether or not using the PI3 kinase delta inhibitor may not be able to rescue the T regulatory phenotype?
Yeah. So I guess the question is really about the Treg and APDS. You know, this is where, you know, I don't have a good knowledge about that. I have to admit, so I find it difficult to answer that question. It is not something that, you know, I've been working with these people. It is not something that has been at the forefront of their thinking. As far as you know, this question is new to me. I'm not aware that, you know, the Tregs are a peculiar problem for these people. Or the delta inhibition will be a peculiar problem. It is not something I have read in the literature or have heard during development of this thing. So maybe the person online has more information than I have, but you know, I cannot answer that one, I'm afraid. Sorry.
Well, again, thank you very much, Dr. Van Hazenbroek, and also thank you very much for all the audiences and with all those great questions. And again, I have to apologize that we do not have time for all the exciting questions, But again, I think this presentation really highlights how important it is to continue understanding the biology, and then the understanding of the biology will facilitate the drug discovery. So with that being said, appreciate everybody's time in this talk, and thank you, Dr. Van Hazenbroek. And also next week, we will have another session of the webinar, and that session will be, just wanted to highlight here. So hopefully everybody can see the slides. So next week, the speaker will be Dr. Shi Chen from Baylor College of Medicine. And then he will talk about stressing out the cells therapy resistant cancers. And some of the talk will be focusing on the KRAS inhibitors and also the recent publications made on how the cancer cells stress make them respond to the different therapies. So please welcome everybody to continue join the webinar and then participate the scientific discussion. And if you have any questions about Medchem Express, our service and products, please email us. Thank you very much.
Thank you. Bye.